Hello to everyone, and welcome to this fifth and last webinar of the webinar series in preparation of the second International BioHackathon of BITS. In this webinar, we are going to conclude our journey in the field of transcriptomics and metagenomics, and in particular, in this webinar, we're going to continue the explanation about how to perform bioinformatics analysis for study microbiome, in particular addressing the topics of preprocessing count data and inferring in bacterial interaction network. So in today's webinar, let me share the presentation. So in today's webinar, the speaker will be me and Piero Marotto, both working at the Department of Information Engineering here at the University of Padova. And we are going to see that the webinar is split in two main parts. In the first part, I will have the pleasure to introduce you to the concept of 16S count data we already saw as the last output of the bioinformatics analysis in the previous webinar. We're going to study a little bit better the characteristics, but also the biases of this data and how we can mitigate them with count preprocessing. In the second part of the webinar held by Piero, on the other hand, you will have the chance to see how from this data you can, through powerful computational method, aim to infer the bacterial interaction network described by your count data. But let's start from our input, the count data. As you saw from the previous webinar, one of the last output of the read preprocessing, when you end up having something that looks like a table, a matrix, where you basically count how many reads you're able to associate to each OTU or RSD, depending on how you run, for example, the CINI pipeline. So on the row, you have your OTU or RSD, or if you already perform the taxonomic association, you may think about having your species, genus, or the level that you prefer to analyze. On the column, on the other hand, you have the different sample that you collect in your study. So the numbers are integer that describe how many reads you were able to associate to that feature, the row, for each sample, the different column. Depending if you're using OTU, RSD, or if you already collect the information about the taxonomical level, while the number of rows or the number of features may range from a few hundred up to some thousand, again, depending also on the different environment you are studying with that sample. In terms of the number of columns, number of samples, this depends a lot on the experimental settings, on what you are studying. You start from studying with dozens of samples up to maybe hundreds of samples if you have time series or larger cohort, up to 1K or even more samples in large consortium studies or large scale studies. The number that you see in that count table, we are going very often to call it count table or auto table, is an approximation of the abundance of the different species that you collect during the sampling process. So you should expect to see some differences in that number, especially between chunk of column that describe different experimental conditions. Indeed, the community composition, so the abundances of the bacteria in a specific sample, is some sort of function of the experimental group of experimental condition. However, even between columns or sample inside the same experimental condition, the same experimental group, you still should expect some variability. This is what is called the biological variability, and it derives from the fact that the colonization of the bacterial species, bacteria grow, the interaction are all stochastic process, so it is expected to see some variability even between samples that are describing somehow the same biological condition. So the raw count matrix, the number of reads that you measure there, is a mix of two signals. A biological signal, the variability you observe in the count value are due to some biological phenomena, but also, and very important for the topic of today's webinar, technical variability. So you observe extra variability in the value, in the numerical integer value that you observe, not because it's describing some biological variability, but because it's additional variability introduced by the experimental procedure, by the sampling of the bacteria during the collection, by the preparation of the biotechnological process to be able the bacteria to be sequenced, and by the sequencing process itself. We are going to see in this webinar that indeed raw count data are affected by many technical biases. The first family of biases is the one that we define as batch effect. Indeed, when you sample, especially when you prepare a large collection of sample, typically you perform the collection of the sample and the sequencing in different batches. That you can think about them as different moments during your experimental procedure, and at each moment the samples you collect are maybe sequenced by different operators or by different sequencer or different time of the day with different humidity and temperature condition. All this batch of sample that you are preparing the same moment define what is a batch, and this the fact that you are analyzing the sample in batches, so when I say analyzing, I mean again extraction of the bacteria, biotechnological preparation of the sample, and sequencing, introduce an unwanted, fully technical variability that depends just on the batch, not on the biology that you are trying to describe and measure with this experiment. Okay, so this is the bias called batch effect. Even if you are in a small experiment when you are able to collect all the sample at the same times, do the extraction of the material with the same kit, the same operator, the same sequencer, so you are within the same batch, even there, some technical biases are present. For example, when you perform sequencing, you probably know that the total number of reads that the sequencer is able to associate to each sample is a fully technical parameter. It's not a function if the bacterial community is rich or not, it's just how you set the sequencer, the number of the read that you see. So roughly speaking, the sum by column of that matrices is the number of read that you assign to each sample. The number of read you can observe in real count matrices is quite variable, so for sure is not constant. In the worst case scenario, you may have even an order of magnitude between one sample and another. Maybe in one sample you have 1 million of reads, in the other you have 10 million of reads. Okay, this is a quite large difference, but still you may think about having 2 times or 3 times or even more the number of reads between one sample and another, and this is just technical ability. It's just due to the sequencing process. This bias, the fact that even the number of reads is not comparable across sample, is what we define as the sequencing depth bias. And this is telling you that if you don't consider this variability, well, the measure that you observe in the count metrics are not very good approximation of the bacterial abundances, because indeed the magnitude of the number that you observe is basically meaningful. It's just a matter of how you set up the sequencer. So just to give you an idea, this is a small toy example of obviously a very extreme case, but again, the purpose is in this example to let you understand the concept. Let's say that we have 3 environments, A, B, and C, with just 3 species, 3 taxa, X, Y, and Z, with that absolute abundances. So in environment A, we have 8 bacteria of type X, 4 bacteria of type Y, and 2 bacteria of type Z, and so on. This is our 2 bacterial abundances. But we know that we cannot measure directly the abundances, we can do only through sequencing. And let's say that you obtain, running the sequencer for the 3 samples that you collect from environment A, B, and C, that amount of read. So as you can see on the bottom, the sum by column, the total number of read, is quite different across sample. And this is, again, realistic. But since you are relying on the absolute abundance, you will see that it seems that now sample 2 is overall half expressed, so there is half of the bacteria in sample 2 compared to sample 1, while in the reality, in the environment A and B, they have the same abundances. And it seems now that in sample C, since you have a comparable amount of read count compared to sample A, you may say, okay, environment C has the same exact composition of environment A, while as you can see on the table on the left, this is not true. Again, this is due to the fact that the read count is not constant among sample, and is just a merely technical effect of using the sequencer with just a different amount of read. So this is telling you that the absolute magnitude of the read count that you observe in the count data is not directly meaningful. Okay? Another source of technical bias, so unwanted technical variability in the value that you observe in a count table, are the one related to the process on how you collect the different sample from the different environment. The biotechnological process that we have nowadays to collect sample from the different site, different ecological niche, from the soil, from our skin, from whatever you're interested in measuring the bacterial composition, these are biotechnological processes that are not error-free, they are not perfect, they have not a 100% efficiency. This is telling you that you should expect to be able to collect not all the species that are truly present in the environment, but also that low abundance species have a high probability to be missed, to be not collected, and in general that all the species, independently from their abundance, you may sample are not very precise representation of them. Again, just to give you an idea, let's see for example, when our environment is represented as an urn, our bacterial community. In this case, with 40 balls, we can think about the overall absolute abundance of bacteria. So we have 50 bacterial cells. And the different colors are again the different species, the different taxa. We saw there this, the abundance, we have 20 bacteria of type 1, 10 bacteria of type 2, and so on. And our extraction protocol, our sampling process, is not perfect, allow us to measure just the 25% overall biological material, in this case just drawing from the urn 10 balls instead of 40. What is happening? Well, on the top of the bottom right part of the slide, you may see a possible sample collection, the results of a possible sample collection. We are able to collect overall 10 bacteria, our 10 balls, but what we observe? Well, that taxa 4 that was expressed with 5 bacteria cells in the original sample, now it seems that is turned off, that bacteria species is not present in our sample. But also other species, for example, in the original sample, taxa 2 is 2 times more abundant, taxa 1 is 2 times more abundant than taxa 2. If you now see that 20 versus 10, it becomes 7 versus 2. So now it seems that taxa 1 is more than 3 times present compared to taxa 2. So this is telling that the internal relative abundances is somehow messed up by the fact that you are sampling a lot less material compared to the real amount of material. So just 10 balls on an urn that contain 40 balls. What about if we consider that we sample more than one community, more than one environment? We have more than one sample, right? Well, we can still use the same idea. Now we have 2 samples described by 2 urns. Overall, the community, the bacterial community had the same amount of bacteria, so 40 overall bacterial cells in both samples, in both urns, with just slightly different internal composition, so internal absolute abundances. For both the urn, for both our environment, we were able to sample, again, just 25% of the raw material, so 10 balls in both urns. What is happening now? So not only in the first sample, again, we completely miss taxa 4, we have a 0 number of balls, so you can think about a 0 number of reads for that taxa. Also, in the previous example, the relative abundances in sample 1 are messed up. We have no more that taxa 1 is 2 times more present than taxa 2, now is 3 or even more times, 7 versus 2 instead of 20 versus 10. But also if we look across between samples, we see now that the relative abundances even between samples are again no more realistic. For example, from the sampling process, it seems that now taxa 3 in urn 1 is just half of the same taxa 3 in urn 2. We see 1 versus 2. But in the original population, it was 5 versus 7. So not a 1/2 ratio, but almost a 1-to-1 ratio. So this is telling you that collected sampling, collection of samples, so a sampling process of the different environment of the different bacterial communities, the less efficient is your extraction protocol, the better is the representation you may think of having in your sample and then in your number of read sequences. So this is telling you that if you just consider the raw count that you observe in a count table in an OTU table, in an ASV table, and so on, this is really not good approximation of the true bacterial abundances of the community. This is also related to not just sample collection, so when you reach your ecological niche and with some biotechnological process you collect the bacteria, but also at the stage of sequencing. Indeed, both bacteria compete somehow to be collected, but also the fragment that you put in the sequencer somehow compete to be attached to the flow cell and then sequenced, so to become reads. So both sample collection and sequencing, we can see them as a competitive sampling process with no replacement and with a limited, fixed, completely arbitrary sampling size. This is telling you that bacteria that are few abundant or fragments that are few abundant, they compete to be captured or sequenced, and they are the one with the highest probability to be missed, meaning that you are not able to capture that bacteria or to ensure that there is at least some read describing that bacteria. This is, again, a huge bias for low abundance species, but as I showed you before, even higher abundance species are still having some issue in terms of relative abundance within a sample and between samples. This is due to what is called the compositional nature of sequencing count data. And this is a characteristic of all sequencing count data. So even single-cell RNA sequencing count or RNA-seq sequencing count. Because again, this is related really to the biotechnological process that modern technology provide us to measure biology. What do we mean with compositional data? Well, there is an example on the right. You should think about two different experimental conditions to sample, to environment, or to, I don't know, cell culture if you're doing single-cell RNA-seq. You have three features, taxa S, taxa A, expressed both with a value of 20, and taxa B that is expressed with 10 bacterial in condition 1 and 60 bacteria in condition 2. So roughly speaking, taxa S and taxa A are uniformly expressed, present in both conditions. It's just taxa B that is overexpressed in condition 2 compared to condition 1. If you think that now you're performing sequencing on this abundance of bacteria, so let's say you're able to just collect 20 read for condition 1 and 20 read for condition 2, well, what you can expect? You can expect that the sequencing, that again is a sampling process without replacement, and where the sampling size, 20 reads, is just a function of the sequencer, is able to preserve the relative abundances. So still, if you check the relative abundance of condition 1, you see that taxa S and taxa A have the same percentage of the overall amount of reads, and are both higher and double the one of taxa B. Same if you go to condition 2, you see that the same amount, the same percentage of read is distributed between taxon S and taxon A, and both of them are a lot lower of the percentage of read assigned to taxa B. So sequencing process is able to preserve more or less relative abundances, but not absolute abundances. If you look on the bottom of the picture, you see that now it seems that taxa B in condition 1 is present as much as taxa S and A in condition 2. So all of them have 4 read assigned. So the fact that relative abundance are more or less preserved is not a guarantee for you that also absolute abundances are preserved. Again, indeed, in this sample, it seems that taxa S and A in condition 1 are more expressed than the same two taxa in condition 2. But we know from the ground truth on the bottom that it's not true. And it seems that now, again, taxa S and A in condition 2 is expressed as much as taxa B in condition 1, while we know from the ground truth that they are 2 times more expressed compared to taxa B. Again, this is because the number of reads that you are measuring thanks to sequencing is just a function, is mainly a function of the absolute abundance, the absolute number of reads, because sequencing count data preserve relative abundances, not absolute abundances. As you can imagine, for us as bioinformaticians, it's quite interesting not only to see difference in relative abundances, but also I would like to know if a taxa A is differentially abundant in two conditions, so the absolute abundance, because maybe it's something that is telling me a very important information about that community. Again, this is, technically speaking, impossible with sequencing count data, because again, they are composition. Compositional data are more frequent than you may expect. Indeed, each time you work with proportion, percentages, or part per million, whatever, these are all compositional data. Compositional data have a very evolving definition. If you just grasp the idea that compositional data are informative in terms of relative abundances, and that the magnitude is just a function of the sampling process, this could be enough for our purposes. More formally, we saw that compositional data stay on the simplex, so we are not free-floating in the Euclidean space, but we are constrained on the simplex. So why this characteristic of data is important? Why the compositionality of data is important for data analysis? Well, because most of the statistical or methodological tools that we used when we did multivariate analysis, correlation, clustering, hypothesis testing, regression, they assume that the data stay on the Euclidean space, not on the simplex. So this is telling you that you cannot use directly naive version of this method. You need to do something more, something that you typically achieve thanks to preprocessing or downstream analysis. So, strictly speaking, compositionality is not a bias. It's just a characteristic, a property of count data. Okay? But it is a characteristic that is the reason why we saw the relative abundance somehow mess up, or why we saw very often a lot of zero. Is one of the reasons why low abundance taxa are the ones that have a higher probability to be missed. Indeed, if you open a OTU table or a count or a SPIEC table, you see that there is a lot of zero entry. Some zero are biological, meaning that that species is not present in that sample, in that environment. Other zero are called technical zero, meaning that you see a zero in the count table, but biology tells you the environment, on the other hand, had that species present. Again, this is due to compositional nature of sequencing data and the fact that with a limited and low sampling depth, sequencing depth, you miss low abundance stuff. This is the well-known phenomenon, if you are familiar with single-cell RNA-seq, of the dropout, where low abundance genes that express very few amount of transcript have a higher probability to be missed, to be not sequenced. This is the same, again, all sequencing count data are composition. And this is another source of bias, the fact that you are missing data. You should know that there are missing data in your count table. So, as you can imagine, these are all sorts of technical noise. The bias that we saw in the presentation are not the full list of technical biases. There is a lot more. And this is strongly suggestive that you absolutely need to do something. Otherwise, you're measuring more technical noise than the true biological signal. This is telling you that if you consider your raw OTU or SPIEC table, this is a very bad approximation of the true community, of the true abundance of the bacterial community that you collect in the different samples. You absolutely need to do some preprocessing. Still, preprocessing is not perfect. Will not allow you to recover the exact absolute abundance of the bacterial community, but still is a lot better approximation compared to using your raw count data. And this is exactly why in bioinformatics we perform different steps within counter processing, mainly a first exploratory analysis when we apply computational method to reduce, to mitigate this technical bias. And then sometimes we also apply some data transformation. Let's see very quickly some of them. When we talk about exploratory analysis, we talk about the same conceptually task that we perform also on RNA-seq, so that you saw in one of the previous webinars. The idea is that you start to inspect your data in two main ways, computing some metrics, some summary statistics that allow you to quantitatively assess some properties of your sample. Or very often you also use some visualization, some plot that allow you to grasp some properties on some biases that you may be present in your data. Very often these statistics, these metrics, these visualization are done both on raw count data to have an initial idea of the level of noise of batch effect or all the bad property that you may have, and then very often after count preprocessing. So to have even a visual expression if something is changing in your data, a hint if your preprocessing is going in the right direction. As you saw in the previous webinar, you can do many exploratory analysis. For example, you can think about computing alpha or beta diversity indices and then plotting them with, box plot for alpha diversity indices. Or use dimensionality reduction techniques like PCOA or NDMS to see in a lower dimensional space all the different samples. This is very common, for example, for beta diversity. All the analysis, again, as you saw in previous webinar, can be done by CINI, most of them. For sure, all of them can be done once you move to R and Python and you install the dedicated bioinformatics packages. During this exploratory analysis, very often you can also perform some filtering, meaning removing feature or sample that you may think contain too much noise to be somehow corrected. Filtering in general, even in RNA-seq, single-cell RNA-seq, is always a trade-off between returning more data with the possibility of having some noise, or reducing the data to analyze and keep only very clean data, but again, losing potentially a lot of information. In terms of 16S count data filtering, there are typical filtering, the prevalence one, when you keep only tags that are expressing at least some percentage or some absolute number of samples, or filtering based on relative absolute abundance, meaning that you keep only tags that are abundant enough, with the idea that low abundant tags maybe have a lot of noise. Or maybe sometimes you remove entire sample if you realize that there are too few widths in that sample. This is the library size filtering. Again, this is not an exhaustive list. These are just the most common ones. Moving to the second substep of counter processing, this is the step when you want to really mitigate, evenly remove, realistically mitigate all the different sorts of technical bias that affect your data. This is done by three main sub-steps: batch effect removal, count normalization, and zero imputation. And the order of these three family of counter processing method, it depends a lot on the specific analysis that you are performing, but also on the assumption of the data. For example, some batch effect correction method assume that the data are already normalized, and so you need to perform first normalization and then batch effect removal. So it depends a lot on the bioinformatics tool that you're using. In general, with batch effect correction, we define all the methods that allow you to try to reduce or to remove the unwanted technical variability that you have in your count table just because you preprocess and produce the data in different batches. There are very few methods specifically developed for 16x count data. I put a couple of examples there, while the majority of batch effect correction methods that are typically used come from other omics sciences, typically transcriptomics, both bulk and single cell. You see there on the left a picture of the different batch effect correction algorithm, that BECA acronym that you saw on the left, divided by the different type macro family of methodologies, and you see that most of them are in blue or in black, that are the column that encode RNA-seq and single-cell RNA-seq. I think most of the methods that you can find in literature are designed for that type of data and just adapted, just used for 16S count data. Another very important step is count normalization, that typically have the goal to address the biases related to different sequencing depth and partially sample composition. Count normalization method typically works by multiplying by a factor, a different scale factor, the different column in your count table. Again, there are not too much method specifically developed to work with 16S count data. The most famous one are the metagenome SPIEC-EASI, the RCSS, and the GMPR method. Again, very often people use a method developed for other kind of omics sciences, typically single-cell RNA-seq and bulk RNA-seq. So it's very often you will see applying the TMM normalization or the jar normalization, even for 16S count data analysis. Again, even if, especially the GMPR method, should to have very good performance in normalizing count data. Last step, very uncommon nowadays, more common in single-cell RNA-seq count preprocessing, less common in 16x count preprocessing is the zero imputation step, when you would like to correct for the sample composition bias and the sparsity bias. So in a nutshell, you would like to distinguish between biological zero and technical zero. And when you spot technical zero, impute the right value that you should observe if you don't have miss that reads that tax. Okay. Even in this field, there are very few methods specifically developed for 16S data. A couple of examples are deep IMB and impute. And even here, most of the methods are just taken from other omics sciences, again, very often from single-cell RNA-seq. All these steps have a practical impact in analysis. There are a lot of comparison studies when people, researchers, tested different batch effect correction, normalization, and zero imputation method, and they proved that they have a huge impact in providing a good or bad quality for downstream analysis, a good or bad pre-processed single-cell count data. This is, for example, a study done by our research group when we test 5 different normalization methods, the one listed on the right, 5 different zero imputation methods, as well as their combination, and we saw the pre-processed count data, how good they are in resembling the true biological abundance in terms of recreating regional sparsity, identifying the right species in terms of present but also relative abundances, how they impact the accuracy of alpha and beta diversity, as well as how they have an impact in the accuracy of differential abundance analysis. And this study, as well as many other studies, proved that a good count per processing, good normalization, zero imputation, filtering, and so on, have a huge impact in the accuracy of downstream analysis. So this is a computational step that you should always take into account very seriously. Last, typically you may want to perform also some transformation of the data. One of the most famous one is the transformation that tries to somehow handle the compositional nature of count data, and it derives from the works of a scientist called Hutchinson that was performed during the 80s. Hutchinson proposed different transformation called log ratio transformation. It's really a family of transformation that, in a nutshell, allow you to map compositional data into Euclidean space. So to move from the simplex to Euclidean space, and so to enable again all the bunch of methodologies, regression, principal component analysis, Euclidean distance that you may want to use on your data. Hutchinson proposed different log ratio transformation. The most famous one is for sure the CLR, the centered log ratio, where for each sample you transform each entry in the count matrix as the log of the row count over a factor that is typically the geometric mean of your sample. Obviously, when you have a lot of zero, the geometric mean will be zero, so you typically add a pseudo count or you perform it only on not zero entry. And even this, even if it is not, let's say, dedicated software like for normalization, zero imputation, you may find a function that computes CLR in a variety of packages that do many other analyses. On the right, you see a few examples of packages and their relative function that allow you to perform the CLR transformation using R programming languages. Hutchinson did do even more, even if you don't want to move your data from compositional, so from the simplex to the Euclidean space, he developed an entire geometry that allows you to perform, for example, distance or normal operation directly in the simplex, but now in a correct way. So this is less common in bioinformatics analysis, but is a very important series of functions that you may want to use even in other research fields. So the take-home message of this first 30 minutes is that if you consider for your downstream analysis your raw count table, so your RSG table, OQ table, or species table, without preprocessing, now you know that is a very bad approximation of the abundance of the bacteria in the environment from which you create that sample. Sequencing step, sample collection introduce a lot of biases, and this is exactly the goal of counter processing, to remove them. It's also very important to be critical in understanding how much a preprocessing step is important, or even if it is important, if it is needed. Indeed, different downstream analysis may benefit more or less from different preprocessing steps. For example, you want to compute an alpha diversity indices, just measuring based on the species abundance, then a zero imputation step is quite important because it's exactly what the alpha diversity index is measuring, the presence or absence of species. On the other hand, alpha diversity indices may be quite, let's say, not affected by counter-normalization, because counter-normalization mainly work on not zero entry, just scaling up. So something that is negligible for alpha diversity indices. You want to perform clustering of a sample maybe based on Euclidean distance, then now you know that for sure you need to perform the CLR transformation, because otherwise you're applying Euclidean distance on data that are not in an Euclidean space. Or you want to perform a differential abundance analysis, and the method that you choose have an internal modeling for the sparsity, the different library size, the composition and nature of the data. So that method was as input raw count data. Because all the preprocessing steps are done somehow internally or are considered by the model of your downstream analysis tool. So always read the assumption of the processing step or the downstream analysis that you want to perform to be sure to perform the best preprocessing for that data. We can say a lot more about the technical biases and how we can mitigate them, but again, these are not exhaustive list, but for sure it contains the most important element in terms of biases and way to recover. And once you have really your preprocessed count stream down 16S count data, is really where somehow the fun begins, because it's now you have clean data that you can provide as input to a bunch of downstream analysis that are now really the goal to extract biological information from your data. And one very important downstream application is the one that now Piero is going to present in the second part of the webinar. So thank you for your attention, and I leave the stage to my colleague Piero.
Hi everyone, I'm Piero Marotto, and I am a PhD student from the University of Padova, and in the second part of this webinar, I would like to present to you the how you can go from the 16S count data, so what preprocessed even, so from where Giacomo left off to a bacterial interaction network. So a very quick recap on what microbial interaction networks are. Here you can see that, as you probably know, microbes don't live in isolation. Instead, they live in a community where they form and they interact with one another. Here is a depiction of a mock community where you can see various types of interaction, from, I would say, more simple interaction like cross-feeding of resources or competition, to also more nuanced interaction like quorum sensing or biofilm coaggregation. And why it is important to know these interaction networks of microbial community? Mainly because if you want to engineer the community, so you want to modify the community to reach a desired output, so maybe in a clinical setting or in an industrial application, you need to know this interaction network, because you need to know the effect of your modification on the rest of the community. So, but why we are talking here today about reverse engineering or inference of microbial interaction network? Because there is a problem, and the problem is that we cannot directly observe microbial interaction networks, at least for large scale communities. And this is why there exists some culture limitation, in the sense that we cannot culture in labs a large proportion of microbes. This is because we cannot replicate the environmental condition needed to grow many of the bacteria that are known. So the solution is inference, to indirectly reconstruct or reverse engineer this interaction network from other observable data. And what are these observable data? Are usually sequencing data. So you start from the 16S count table like Giacomo presented to you. So you have your bacteria or features in the rows, and the different samples you collected on the columns, and where each cell, each entry in this table represent the measured abundances of the bacteria in the different samples. Note that usually the number of features, so the number of bacteria that you see, is much greater than the number of samples that you collect. So this is your observable input data, and what you want to obtain is the reconstructed interaction network, which can be represented as a graph, which is a mathematical structure, very popular mathematical structure, in which we have nodes and edges. The nodes are, in the context of microbial interaction network, the different members of the community, so the various bacteria that you have in your community, and the edges are the inferred interaction between them. So at a very high level, how does these inference procedures work? Let's think of a simple mock example where we have two species, X and Y, and we can say, and we know that Y positively interacts with X, and we also know that Y feeds off an unknown resource that we can call A. Then you can take a look at one observation of this community. You see that the resource A has a certain level, consequently Y has a certain abundance, and X has a certain abundance. Then you look at another sample where you can see that the abundance of A, of the resource, increased slightly. This in turn increased the abundance of Y, since Y feeds off of the resource A. And in turn, this determined an increase in the presence of the species X, because Y positively interacts with X. So if then you take many more observations of this small community, you will end up with something that looks like this. And you can see that the main idea behind concurrency inference is to note that since the two species positively interacts, their abundances covary across samples, across observation, in the sense that if Y increases or decreases, also the abundance of X decreases or increases accordingly. Of course, this covariance is not linear and is not free of noise. For example, in some sample, one of the two may not be present, but the general idea is that if the two species interact between each other, there will be some sort of covariance in their abundance across samples. So what we see here, the two histograms are none other than two rows of the count table of our input count table, and the general idea in concurrency inference is that you take pairs of rows, so pairs of bacteria, of features, and you measure some form of statistical association between them, so you obtain a number that we can call alpha. Of course, then you need to assess if this association is statistically significant, because of course, as you know, if you take a form, if we measure some statistical significance, some statistical association between two random vectors, you will get a non-zero value. So you need to determine if the value that you obtain is significant. If the value is significant, you can place an edge in the inferred network. So in this case, the black edge between the two bacteria that we saw before. So what is the most intuitive and most, I would say, simple way to measure this association is the use of correlation. There are, as you probably know, many types of correlation, from Pearson correlation to Spearman or Kendall-Stau correlation. So the idea is that you take every pair of rows in the count table that are two vectors X and Y, like we saw before, and you compute the correlation value, which is what you can call R, and which is like what we called before alpha. And then you need to determine the significance of this value R. You can, for example, there are various ways to do this. You can, for example, choose a threshold, and every a correlation that is greater in magnitude compared to this threshold is determined to be significant. Of course, this is an arbitrary choice of a threshold, which is not generally a good idea. The other option is to use the p-value. So with correlation, there are a lot of theory about this. You can use, for example, parametric tests like t-test for Pearson correlation. But note that, as the name say, the parametric test makes some parametric assumption about the variables on which we are measuring correlation. And note that usually these assumptions are not valid in the metagenomics field. Or the other option is to use a permutation test. So this is a test that makes no assumption, so it's always valid, but it is more computationally expensive. But why the fruit with a bitter taste? Because there is a problem, as you saw before, and as Giacomo told you before, sequencing data are compositional, and so I will not go over it again because Giacomo very nicely presented it to you before. So think, for example, of 3 samples in which 2 species are constant, so they are always 5 absolute abundance, but 1 species increases in the 3 samples. Of course, you in metagenomics field, you need to look at the proportion because the sequenced totals are arbitrary, so you need to normalize your samples, so you end up with relative abundances, which is something like this. But then if you measure correlation between the species on the relative abundances, for example, the correlation between orange bacteria and the green bacteria, you will end up with, in this case, for example, a perfect anti-correlation, which is not real in the sense because in the absolute data, the green taxon is constant, so it does not vary. So as for definition, it is not correlated with anything. Correlation is not even defined because it does not covary. But if you measure it on the relative abundance, you would see a spurious correlation between the two. So you could end up inferring an interaction between them even if it is not present. So one of the possible solutions to this, or at least a partial solution to this, is to use log transformation, as Giacomo told you before. So why they can help? Because log ratio transformation are scale invariant in the sense that the CLR, for example, computed on the absolute abundances is equal to the CLR computed on the relative abundances. But be warned that the log ratio transformation do not necessarily preserve correlation between the correlation structure, meaning that the correlation between two variables x and y in absolute abundances is not necessarily and often is not the same as the correlation computed on the CLR transformed variables. So one of the methods that was developed ad hoc for the inference of microbial interaction network that uses correlation is CICI-LASSO. I will go over it very, very quickly, but just to let you know. So in input, you have your 16S count table. Note that CICI-LASSO expects that the columns are the features and the rows are the samples. So you need to usually transpose your input table. Then CICI-LASSO applies a log transformation plus a pseudo count to the input table. And then it applies a lasso estimation of the covariance matrix of the variables, and it also selects the optimal regularization parameter lambda. Very quickly, this is, I would say, a very schematic formula what CICI-LASSO does. So it estimates the correlation matrix using a maximum likelihood estimation, and it adds a regularization parameter which enforces a sparsity, so it enforces 0 values in the correlation structure. The output then of CICI-LASSO is a list where you can retrieve the various output that you would want to know to infer the interaction network. So, for example, the correlation matrix then you can use as an interaction network or a reconstructed interaction network. This is the command that you need to run to execute CICI-LASSO. So you have your input, which is the count table. Remember that you need to often transpose it. There is a flag to say to CICI-LASSO if the input table are counts or if you have already normalized it, so to proportions. Then you can also tell the pseudo count value that you want to use. You can specify the number of folds in the cross validation to select the optimal lambda parameter. You can also specify the extreme of the lambda path, in the sense that, of course, CICI-LASSO does not test all the possible real numbers to select the optimal lambda value. It searches in an interval with some discrete values, so you need to tell CICI-LASSO how big you want this interval to be and how many values in this interval you want to test. But there is also another problem with using correlation, which is the inference of spurious edges. Let's think for a moment at a very simple mock example in which we have 3 species, X, Y, and Z. And the interaction structure of these 3 species is, I would say, very simple. X positively interacts with Y, and Y positively interacts with Z. But if you then do a 16x experiment, so you obtain a 16x table from this community, you would see a covariance, a positive covariance of the all three species across the samples, meaning that if X increases, both Y and Z increases over the samples. So if you try to apply correlation to reconstruct the interaction network from this input table, you will end up with an interaction network that is something like this, where all species positively interact between themselves, and there is also a positive interaction between X and Z, which is not really present because X and Y do not interact in the real interaction network, but it is inferred due to indirect effect of correlation. So the solution to avoid this kind of spurious interaction is to use the concept of conditional independence. Conditional independence allows you to isolate the direct association and avoid the use of the introduction of spurious associations. Mathematically, the definition of conditional independence is that two variables, x and y, are conditionally independent if the probability distribution of x conditioned on y and all the other variables is equal to the probability distribution of x conditioned only on the other variables. What does this mean? It means that y does not give any additional information in explaining the variance of x. So looking at our previous examples, we can say that x and z are conditionally independent because the probability distribution of z conditioned on x and y is equal to the probability distribution conditioned only on y, meaning that x gives no additional information in explaining the variability of z. And so if you are able to reconstruct the conditional independence structure of the variables, you could end up with the true interaction network, so without placing an edge between x and z. But how can we exploit this conditional independence framework to infer our interaction network? We can do it because there is a neat relationship within the precision matrix, which is the inverse of the covariance matrix, of multivariate normally distributed variables and the conditional independence condition. This relationship is that if you have again multivariate normally distributed variables, a zero entry in the precision matrix implies a conditional independence between the two variables. So if you somehow can know this precision matrix under the assumption of multivariate normality, you could know the conditional independence structure of your variables. And how can we estimate this precision matrix? Usually, given an IID observation, we can estimate the precision matrix using a maximum likelihood estimation with a regularization parameter lambda. This is because the problem is ill-posed, because usually, as we saw before, you have much less samples compared to the number of features that you want to estimate. So you need to add a regularization parameter. This is the formula. So you have the log likelihood as the first term, and the regularization parameter that imposes a sparsity on the precision matrix. To solve this problem, there are many algorithms that have been developed over the years to efficiently solve this problem. Two of the most used are the MB algorithm and the GLASO algorithm. But here again, we have a problem in that it is how can we select the optimal regularization parameter lambda. In the field of metagenomics and microbial interaction network inference, one of the methods that is used to select this problem is the STARS algorithm. Very quickly, how does it work? So you start from choosing some values of lambda to test, which is your lambda path, the same as we saw for CCLAS before. So you end up with a series of discrete values of lambda that you want to test. Then for each value of lambda, you create B subsample of your data. So you take your input count table and you subsample it to obtain B tables from the original one. Then on each of these tables, you apply your inference of the precision matrix. So you end up with B precision matrices, which can be interpreted also as a graph, as we saw before. Then you compute the edge instability of each edge. What is the edge instability? The edge instability is the fraction of subsamples in which an edge takes its least common state. what does it mean? For example, looking at this small example, for example, looking at the edge between green and purple, we can see that in 3 of the subsamples, the edge is present, so it is inferred to be present, while in one of the subsample, the edge is not present. So in this case, the least common state is the edge not present, so this case over here, and the frequency of the least common state is 1/4, so 0.25. In the other cases, the edge instability is 0 because all the subsample agree that there should be an edge between the 2 nodes. Then you can compute the average edge instability, so you compute the average of all the values that you end up over here. So you end up with one value of average edge instability for every value of lambda. And then you select the optimal lambda value as the one that guarantees an average edge instability that is lower than a specific threshold that you need to set, which is often called beta. Why? Of course, this is also a problem because you need to set an arbitrary threshold, but it has the advantage that you can interpret this threshold. It means that on average, your edge instability with this lambda value will be lower than a threshold. So some key takeaways, especially about the selection of the lambda parameters. Since lambda is the regularization parameter, it determines the sparsity or density of the inferred network. Why is that? Because if you have a high lambda value, so a high penalty, it means that the estimation algorithm will prefer to place zeros in the precision matrix to avoid the penalty imposed by lambda. So you will end up with a more sparse network. It has more a more sparse inferred network. While if the value of lambda is lower, so the penalty is lower, you will end up with a higher density network, so a lower sparsity network. The other key point is that using stars, the lambda parameter value is determined by the instability threshold beta. So if you set a lower value of beta, you allow for lower instability. To guarantee this lower instability, the estimation algorithm will prefer a higher lambda, so it will prefer to put a 0 in the precision metric instead of, let's say, incurring the risk of making some mistakes, and this will lead to a higher sparsity. So lower beta, higher sparsity. Instead, if you allow for a higher beta threshold, so you allow for higher average edge instability, this will allow the estimation to select a lower value of lambda, which will in turn result in a lower average sparsity and in a more dense network. Then a very quick observation, the fact that, as we said before, the lambda value are selected along a discrete path. So the more values you put in this path, the more likely you are to end up near or selecting the true optimal parameters. Of course, this is not a guarantee, but it is less likely that you will skip over the true optimal parameter just because of discrete steps. And but remember that the more values that you test of lambda, the higher the computational cost. So let's take a look at some methods that employ this conditional independence paradigm to infer Machiavelli interaction network. One of the first that was developed and is most likely one of the most famous is SPIEC-EASI. How does it work? It starts from an input in count table, and remember that also as a CC lasso, it expects features to this column, so you will need to transpose your count table, most likely. Then SPIEC-EASI applies a CRL transformation with the addition of a pseudo count to avoid the problem with the geometric mean. Then it applies the GLasso or MB algorithm plus stars algorithm to estimate the precision metrics, and it outputs I would say I should add that all the methods that I presented to you today are implemented in R. So SPIEC-EASI returns a custom S3 R object, which is, I would say, quite complicated. And to obtain the relevant output that you want to then look at, so the inferred interaction network, you need to use some specific functions to extract them from the output object, which in this case is called X. So for example, to recover the adjacency matrix, which is I would say, your interaction network, you need to use the getRefit function on the output object X. So a quick look at the function of SPIEC-EASI. This is your input. Remember that you need to transpose it. Then you can select the estimation method, so GLASSO or MB or the MB method. You can also choose the selection criterion for the lambda parameter. I would suggest you to leave it at stars. You can also increase the number of cores to be used if you have more than one. This will, of course, increase your speed, increase the speed of your computation. Then you can select the number of lambda values that you want to test that are in the lambda path. And one of the more important parameters is the parameter thresh, which is the instability threshold that we saw before. Another partial correlation method or conditional independence method is SPRING, which is, I would say, quite similar to SPIEC-EASI, but it has some implementation differences that I will not go into this webinar, but you can take a look at the original paper if you are curious. So the input is as for SPIEC-EASI, it is a table where you need to have features as columns. So you will need to transpose your table. Then it applies a modified CLR transformation to the input table. And it then estimates the precision metrics using the MB algorithm, plus using also a semi-parametric covariance matrix as the starting point. The output of SPRING is a data frame in which you can find all the relevant outputs that you can want to take a look at as by looking at the different fields of the data frame. Here is the function to run SPRING. Of course, remember again to transpose your input table. Then SPRING has a flag because SPRING also has an algorithm to infer on non-compositional data. So there is a flag to say if the data is quantitative, so non-compositional or not. Usually you will need to leave this at false because you will almost certainly work with compositional data. Then there is a quite obscure, I would say, parameter, which is R method, which is the method used to compute the semi-parametric covariance matrix. There are two values for this parameter, original or approx. Approx is much faster, and it usually the results does not vary much between original and approx. So I would suggest you to leave it at approx if you want to use a SPRING. Then it is also SPRING allows you to set the instability threshold parameter, which is called thresh. And also it allows you to increase the number of cores used in the computation. So you can increase this if you want to speed up your computation. And that was it. Thank you very much for your attention. If you have any questions, please feel free to drop them in the chat. Maybe we can wait just a few moments to see if there are any questions. I don't see any, so in case I will just leave you with some last information. In case you have some question, of course, leave them in the chat while I tell you this last info. This was our last seminar. The next step is the BioHackathon. The BioHackathon will take place in Padova on site in the afternoon of the 26th of May, approximately from 2:30 p.m. to 6 p.m. to 6:30 p.m. And on the morning of the following day, the 27th of May, approximately from 8:30 to 12:30. Remember that if you want to attend, you need to register. If the registration form for the BioHackathon is different from the registration form that you filled in to enlist in the webinars mailing list. So if you have only filled in one form and you want to attend to the BioHackathon, please fill in this other form. Here is the QR code to the BioHackathon registration form. If you want to take a look at some other info about the BioHackathon, here it is the QR code to the BioHackathon website. And as a final note, remember that everyone that can take part in the biohackathon, even if you do not attend if you are not attending the BITS conference. So if you are in Padova in the 26th and 27th of May and you are free, feel free to come to the biohackathon. And that was it. I don't see any other question in the chat. So I will see you in Padua for the biohackathon, and have a nice day.